day, everyone. I am Rosandrella Salahay, a second year BSED Science student.
Now, let us know what is the cell cycle and cell divisions and its functions. So what is a cell cycle? The cell cycle is a four-stage process in which the cell increases in size, which is the GOG1 or G1 stage. It is the one who copies its DNA or the synthesis or S stage, prepares to divide GOP2 or G2 stage, and divides mitosis or M stage. The stages with G1, S, and G2 make up interphase, which accounts for the span between cell divisions. In the cell cycle, the interphase is the cell prepares for division. Both the animal cell and the plant cells are their DNA replicated, their organelles replicated, and their cells increases in size. The mitosis and its functions. The mitosis is the process of cell division which results in the production of two daughter cells from a single parent cell. The daughter cells are identical to one another and to the original parent cell. The mitosis can be divided in two stages: the prophase, metaphase, anaphase, telophase, and cytokinesis. In the prophase cycle, the cell prepares for nuclear division. In the animal and plant cell, the DNA coils into chromosomes. Next is the metaphase. In the metaphase, the cell prepares chromosomes for division. Both the animal cell and plant cells chromosomes lined up at the center of the cell, and spindle fibers attached from daughter cells to chromosomes at the centromere. Anaphase. In the anaphase, the chromosomes divide. The spindle's fibers pull chromosomes apart, and one half of each chromosome, called the chromatid, moves to each daughter cell. In the telophase cytokinesis, the cytoplasms divides. Both the animal cell and plant cells, their DNA spreads out to nuclear form. In the cytokinesis, the cell membrane pinches in to form the two new daughter cells. Now let us know what is the meiosis and its functions. The meiosis is the type of cell division by which the gametes, 6 cells, eggs, and sperms are produced. One parent cell produces 4 daughter cells. The daughter cells have half the number of chromosomes found in the original parent cell. During meiosis, the DNA replicates once but the nucleus divides twice. There are two parts of meiosis, the meiosis 1 and the meiosis 2. The meiosis 1 is the first division of meiosis. In the meiosis 1, the prophase 1, each chromosome duplicates and remains closely associated. These are called sister chromatids. Homologous chromosomes will cross over. While in metaphase 1, the chromosomes will pairs align at the middle of the cell. And in the anaphase 1, chromosome pairs are pulled apart with sister chromatids remaining together. Lastly, in the telophase 1, two daughter cells are formed with each other daughter containing only one chromosome of the chromosome pair. As you can see in the prophase 1, the chromatin begins to condense following the interphase, and synapses align homologs and chromosomes shorten. In the middle prophase 1, the chiasmata becomes evident, and in the late prophase 1, the coiling and shortening of the chromosomes continue. In the metaphase 1, the chromosomes line up on the equatorial metaphase plate, and then goes to the anaphase 1, which the homologous chromosomes move to the opposite poles of the cell. Lastly is the telophase 1, which the chromosomes gather into nuclei and the original cell divides. Number 2 is the second divisions of meiosis. So, the second division of meiosis, the prophase 2, the DNA does not replicate. The cell prepares for meiosis 2. In the metaphase 2, the chromosomes line up in the middle of the cell, while in the anaphase 2, centromeres divide and sister chromatids move separately to each pole of the cell. While in the telophase 2, 4 nuclei begin to form and the 2 cells begin to pinch into 4 cells. And in the cytokinesis, cell division is complete and 4 haploid daughter cells are formed. This is the prophase 2, which the chromosomes condense again following a brief interphase in which DNA does not replicate. While in the metaphase 2, the kinetochores of the paired chromatids lined up across the equator of each cell. And in the anaphase 2, the chromatids of the chromosomes finally separate, becoming chromosomes in their own right and are pulled to opposite poles. Lastly, in the telophase 2, the chromosomes gather into nuclei and the cells divide, which is the product of meiosis. Each of the 4 cells has a nucleus with a haploid number of chromosomes. Now, let us know what is the difference between the mitosis and the meiosis. The mitosis has the 4 stages in total, plus interphase. It happens in somatic cells. Its purpose is cellular proliferation and produces 2 diploid daughter cells. Its chromosome number remains the same and its genetic variation doesn't change. While in the meiosis, it composed of 8 stages in total, plus interphase. It happens in germ cells. Its mainly purpose is sexual reproduction. It produces 4 haploid daughter cells and its chromosome number is halved in each daughter cells. Lastly, its genetic variation increased. While there are similarities, it produced new cells, similar basic steps, and start with a single parent cell.
It's caught in the crossfire. It's caught in the crossfire.